So today I'll give an overview of my lab's work on the topic of the origin of life, and we want to understand how this this existential question essentially we want to answer this question. And there's before I get started, I just want to acknowledge that what I'm about to present is drawing from ideas and writings of many people, and trying to integrate them into one experimental research program. So I've just shown you some faces and names here of people that have inspired me, and I'd like to particularly point to Harold Morowitz, who passed away in 2016, to Eric Smith, his collaborator, and to Bill Martin, who's my collaborator. Their writings in particular really influenced the way I think about this problem, and I encourage you to read their work. All right, so let me get started. So the origin of life is a problem of, depending on who you ask, you will get very different answers to this question. To some researchers, the origin of life is a problem of information specifically in the form of what sequence a particular polymer might have of its monomer units. That's one type of information, and one type of way of looking at the problem. Some people will say that self-replication is what the origin of life really is about, and they'd like to see RNA perhaps copy itself. Others will say it's a problem of chemical synthesis, that we have to make molecules because they weren't complex enough on their own on the early Earth, and that's certainly true. Others will say it's a problem of compartmentalization. We have to find some way of keeping organic molecules concentrated and distinct from their environment in order to have life become the way it is, cellularized as we know it today. And of course, all of those things have to be true, right? I mean, all of those things happen at some point in some context. But there is some aspect of this problem, I think, that gets overlooked, that ties all of these things together, that has to tie them all together, and that is self-organization. I think that is really the guiding principle that we have to keep in mind for the origin of life. I'll just try to make the case for that. So, first thing I think we have to ask ourselves, we know that life isn't a thing. It's a dynamic system. It's a process. It's not a static entity. And so the first thing we should ask ourselves is, how do dynamic systems spontaneously form order in science in general? And so let's take an example of a hurricane. This is a problem that's been studied in many contexts. A hurricane is made of air and water, but air and water alone are not a hurricane. We all know that. To get a hurricane, you have to apply an energetic stress, which in this case is a temperature gradient, and there are constraints on what that on how that can be applied. If you apply a gradient that is too small, nothing happens. If you apply a gradient that's too large, you can get chaotic behavior. So you need the right gradient, and then also you need a certain distance scale. Hurricanes don't usually form on the 1 meter distance scale. They usually form on the 1,000-kilometer scale or so. So you have to get the constraints right, as well as the energetic stress acting on or being coupled to your building blocks in order to get this self-organized behavior. So in analogy, if you want to have chemistry that self-organizes and becomes more complex on its own, we have to have our building blocks, which presumably would be geochemicals that would be present on the early Earth. We're going to have to apply an energetic stress, and we don't know what that is a priori. I will argue in the next slide it's probably redox gradients and maybe pH gradients as well. It could be other things though, we don't know for sure. And when we have to think about what the constraints can be. How can we constrain chemistry? Well, that could be through catalysts, which will allow some reaction channels to occur more easily than others. That could be things like natural metals or minerals in the environment, or pH gradients with acid-base catalysis. We could also think about other constraints like scale, right? Just like with the hurricane. Life is cellularized, it's usually on the micron scale, so maybe we could think about scale as well. We could think about conditions like pH and temperature and things like that. So there's lots of constraints we could think about, and that's the way I view the problem. Essentially, it's deciding what pattern, dynamic pattern we want to see, and then deciding what are the energetic stresses and constraints that will allow us to achieve that pattern acting on a set of building blocks. So now let's think about how life works. So first I'll give you the analogy of a fountain. And a fountain is not a thing either. It's a It's a dynamic steady state, right? We have a pump which burns fuel that drives the water up through a channel, and that process is thermodynamically favorable because we're burning the fuel. At the same time, we have the action of gravity pulling down the water and lowering the gravitational potential of the water. And that's a favorable process too, because the water prefers to be at a lower gravitational potential. So these are two processes that look like the rough reverses of each other, but they're not. They're microscopic reverses of each other. They have different thermodynamic driving forces. And when you superimpose those two things, you get the dynamic process that is the fountain. Now, in chemistry and in biochemistry, we have the same thing. We start with geochemicals. Every ecosystem has to do this. It starts with simple geochemicals. It builds from those. It reduces them to keto acids and other carboxylic acids, and those get converted to sugar phosphates, amino acids, and to lipids. And sugar phosphates and the amino acids can react further. They can make with other metabolites to make small molecule cofactors, which can feed back on those processes as catalysts or co-catalysts. They can also react to make the ribonucleotides, which then polymerize to give us RNA. They also are the ribonucleotide triphosphates are also the precursors to DNA, or the amino acids can polymerize and give us proteins. So there is this specific flow of matter in life in this synthetic direction. That synthetic direction is driven by reducing agents and by phosphorylating agents, which are derived from pH gradients across membranes. At the same time, we also have breakdown of these compounds, and those are largely driven by oxidizing agents and by hydrolysis by water. So again, we have two processes that roughly look like the reverse of each other. They're superimposed processes. They're both happening at the same time, but they're driven by different thermodynamic driving forces, and therefore they're not the microscopic reverses of each other. But we really have to have this dynamic system and interplay in place to get something that we would call life. So now let's ask the question, how does complexity emerge in general? The field of complex systems asks this question all the time, and it's studied many different systems, all sorts of things, how birds flock, how swarms behavior. I like the example from computer science, Conway's game of life, right? This is essentially just a black and white grid, pixels. The pixels could be black or white. And the programmer applies a time-dependent series of rules, which causes one pixel to go from black to white or vice versa for a certain amount of time, depending on the environment that it's in. And you write a small number of these rules, and when they are combined, certain combinations of rules will give you really complex, interesting behavior like what you see on the screen right now. So if we were to think about the same thing in chemistry, how would we get a complex system emerging from simple chemical mechanisms? Well, we'd imagine those mechanisms could work together, a small number of them, to create a reaction network that is greater than the sum of its parts, that displays complex behavior as well. Those mechanisms would be dictated by the inherent reactivity of the molecules involved and by catalysts that were available in the environment. Like I said before, maybe these could be something like acids or bases that are present in the environment, or transition metals, or metal ions, or different things like this. So now we have a way of thinking about the problem. What are those? Why would the whole be greater than the sum of its parts in chemistry? There are things you can access with networks of reactions that you can't access with individual reactions happening one at a time. And one of the most interesting things that you see in chemistry is autocatalysis. It's a self-reinforcing feedback that we see in chemistry. A simple way of expressing this is that you have a molecule A plus that reacts with B, and that goes to 2B. So essentially, B is, you could subtract it from both sides. It's a catalyst for the reaction. It's present in both the starting materials and the products, so it's not consumed in the reaction, but it's also a product of the reaction. So this is autocatalysis. I prefer to think about this in a more network-like way, because being an organic chemist. And so imagine you have a molecule that's A, it reacts with some reagents, gives you B, B reacts with some reagents to give you C, C reacts with some reagents to give you D, but then D is big enough that it can split into two and give you back one molecule of A and one molecule of B. Now in this network, you double the number of molecules in the network with every turn of the cycle, and we call this network autocatalysis. Of course, there are side reactions. This is in chemistry. You rarely get a perfect clean reaction, and so that would cause intermediates to leak off of this cycle. You can compensate for that by inputting reagents into the cycle, and if the flux into the cycle is faster than the flux off of it, well, then the cycle can sustain. It can provide all the building blocks you need for other chemistry. Now, there's a nice review I can direct you to at the bottom that was recently written by my colleagues in Paris on the universal motifs. Turns out you can classify all autocatalytic systems into a limited set of motifs. I encourage you to check that paper out. Now, the next question we have to address when we go into prebiotic chemistry is if we had a self-organized chemistry, which I think we had to have had to get to life as we know it, there must have been changes to that original chemistry with the chemistry that we have now. But how drastic could these changes have been? And in general, in the field of complex systems, I think Herb Simon's an interesting thinker in that regard. That when you get hierarchies that develop within a complex system, the hierarchies to some extent lock in what the processes that happen at the levels of hierarchy below them. They don't necessarily make it impossible to change a little bit, but drastic change is quite difficult because those processes support all of the higher level operations. So I think for that reason alone, I make the analogy to the card tower. It's hard to take a card tower off the bottom of the deck. You can take it pretty easily off the top layer, but taking it right off the bottom is hard because those levels support the levels above. And for the same reason, I think that fundamental aspects of whatever self-organized chemistry gave rise to these higher levels of properties, some aspects like the energetic aspects and some of the reactivity would be difficult to fundamentally change. So I think it's fair to think that we can actually learn a little bit about prebiotic chemistry just from studying how biochemistry works today. Now, there's people that completely disagree with this, but as far as we know from studying from our phylogenetic studies that get us quite back, maybe 4 billion years or so, between 3.5 and 4 billion years in terms of what organisms were doing back then, it seems their biochemistry was remarkably similar to the ones that we know today. So I think from that alone, we have no evidence that it is ever otherwise. So I would say that the parsimonious view is to think that there was some similarities between the first self-organized prebiotic chemistry and biochemistry as we know it today. So with that in mind, we can consult the literature on phylogenetics of early biochemistry and rely on some ideas of people like Bill Martin and like Harold Morowitz to think about what would the earliest biochemical pathways have been, and potentially things that are still layovers from prebiotic chemistry. And we put together this map which builds on those ideas, and essentially we could think of it as the CO2 fixing pathways that convert carbon dioxide into small organics, like I mentioned on that bottom rung of that pyramid. That gets us into something called the reverse Krebs cycle, or there's analogs of this that are similar, that produce these building blocks that go on to make all of the other pathways possible. So for instance, pyruvate is the starting point for a pathway called gluconeogenesis that makes all the sugars, which are actually formed as sugar phosphates. And 3 keto acids on that cycle are the starting points for amino acid biosynthesis, and all the other amino acids are derived from those. And then amino acids and those sugars can react, or sugar phosphates react together to give us ribonucleotides. Those can then polymerize to give us RNA. They can react further to give us the deoxyribonucleotides, and then you go towards higher levels of complexity and eventually a genetic code from there. Now, that's skipping over a lot of stuff, but I think that's the rough outline of the kind of consensus thinking on what pathways might have been operative in early biochemistry. And I'm going to test, we want to test to see if these pathways could also have been prebiotic chemistry, if they could have been remnants of the original self-organized chemistry that gave rise to these things in the first place. Okay, so we can test this in the lab, and this is what we've tried to do. The idea is if we look at each of these pathways individually, we'll see if, first of all, we can get them to work at all without enzymes. And then we can look at the different pathways that we studied in the lab and see if we can draw out any commonalities from all of them. And that will give us clues about what sort of conditions, energy, and constraints may have been needed to be applied in the first place to generate that first self-organized chemistry. Okay, so that's what we've been doing. It's a big project. I'll give you an overview of our progress on it. So I'm going to tell you now about the acetyl-CoA pathway, and this is a unique CO2 fixing pathway. It's unique for a couple of reasons. There's only 6, arguably 7 CO2 fixing pathways known in autotrophic organisms, and it's unique amongst those 6 for a number of reasons. First of all, it's the only one that's a short pathway, linear pathway, rather than being a big loop with many reactions in it. It's the only pathway of those 6 that starts uniquely from CO2. You don't need any pre-existing organic in principle to make it function. Phylogenetic studies have shown that it's present in both the bacteria and the archaea, and probably present in the last universal common ancestor. So could this be prebiotic chemistry? Let me mention the chemical mechanism. I know many of you are not chemists, but basically what happens is you take one molecule of CO2, reduce it down to carbon monoxide. This is all happening on cofactors that are based on transition metals in enzymes, things like nickel and cobalt. This one's happening on nickel. CO2 then gets reduced to a formal group that happens on organic cofactors, and then it gets reduced eventually to a methyl group, which is bound to cobalt, another metal. Those 2 groups then combine, forming a new carbon-carbon bond, and you get something called an acetyl group. It's initially bound to nickel, but then it gets displaced by a thiol, and you get something called known as a thioester, acetyl-CoA. The next step from there is that we have a reductive carboxylation, and that occurs on with a cofactor that's based on iron, and you get another carbon-carbon bond. This gives us to pyruvate, which is the precursor to sugars and part of the reverse Krebs cycle. So can that chemistry happen in the lab? And we started this question, started to answer this question in a very naive way. We simply took different metals that were known to be involved in the enzymes and cofactors of this pathway. We took them in their 0-valent forms, so their most reduced forms. We put them into water, we added CO2, we just heated. And we saw, in the case specifically of iron, nickel, and cobalt, we saw CC bond formation, and we saw it all the way to pyruvate. And in fact, of the organics that we analyzed, at least the ones we could see by NMR, we see things that correspond very closely to the intermediates and end products of that pathway. We see methanol, we see formate, we see methanol, we see acetate and pyruvate, shown in red. So then we stuck with iron just for the rest of those studies, because iron is the most abundant metal in the Earth's core and in meteorites. And so we then looked at different temperatures, and it turns out we didn't need to use 100 degrees as we had initially started with. It turns out the reaction can happen even at 30 degrees Celsius. So really physiological temperatures for us, it doesn't need to be an extreme temperature. And then we varied the pressure of CO2, and we went down all the way to 1 bar, so just 1 atmosphere of CO2. And at that point, the reaction is even more selective for acetate and for pyruvate, the C2 and C3 products. And we see very little formate under those conditions. So this chemistry looks a lot like the acetyl-CoA pathway, and all it required was putting metallic iron into water. So after we published that paper, I got an email from Bill Martin, who I hadn't met at that point. And but of course I'd read his papers. He was very excited about that result, as I suspected he would be, because he had been writing for over a decade about that pathway likely having been prebiotic chemistry. And he was trying to do something similar in his lab with his PhD student Martina Preiner and his collaborator Harun Tusuz. They were trying to use hydrogen to drive this chemistry. And so he was very excited when our paper came out. We arranged a meeting, and we decided to work together to see if we could develop a version of this chemistry that was running from hydrogen gas rather than from metals as their source of electrons. And of course, this is more like the organisms that actually run this pathway. They use hydrogen gas as their source of electrons. So we worked together, and indeed, this is possible, this chemistry. Same thing happens under hydrogen gas. You need a higher temperature in this case for 3 different minerals that we identified that do this chemistry. One is magnetite, just rust. Another is awaruite, which is an intermetallic compound based on iron and nickel. And the last one is greigite, which is an iron sulfur mineral. In more recent work that's predominantly done by Haroon Toussouz's lab and working together with Bill Martin, they have found that you can make the same reaction occur over nickel at room temperature now. So we can do this with hydrogen in water at room temperature and mimic the same products and intermediates of this metabolic pathway. Now I'd like to tell you about a second CO2 fixing pathway out of those 6. It's called the reverse Krebs cycle, or the RTCA cycle. And this one's important for 2 reasons. The first is that the compounds that are on it, 5 of them, are the kind of universal building blocks, as Harold Moritz would describe them, for the rest of biochemistry. So you can make all lipids from acetyl-CoA, you can make all sugars from pyruvate or from oxaloacetate. The amino acids are made from pyruvate, oxaloacetate, or alpha-ketoglutarate, and some cofactors are made starting from succinate. So essentially you can make all of biochemistry from the molecules that are on the cycle. It's the only cycle with that property. Some versions of this cycle are autocatalytic in its complete form. That means that, as I mentioned before, that you start off with something that's 2 carbons, acetyl-CoA, you build until you get to oxaloacetate, which is 4 carbons. You build further until you get to citrate, that is 6 carbons, and then citrate fragments into, gives you back 1 molecule, 1 acetyl group and oxaloacetate. So you double the number of molecules with every turn of the cycle. So if a full cycle of this would be possible in prebiotic chemistry, that would be interesting, because it would allow matter and energy to be focused into a small number of building blocks that just happen to be the precursors for everything. It may not have been autocatalytic. It may have been just a linear pathway and only a partial version of the cycle. That is debated. We don't know, so we'd have to explore that. 5 of those 11 steps require ATP though, and 6 do not. So in our initial work on this project, we decided to try to recreate a partial version of the RTCA cycle, focusing on the ATP independent steps because those are simpler to deal with. And so in our first work, which is actually our first paper in this field, we showed that you could actually carry out the bottom, what I call the bottom sequence of this. So the reduction of oxaloacetate to malate, the dehydration from malate to fumarate, and the reduction of fumarate to succinate. That could be done using metallic iron again as the source of electrons, and zinc 2 as a catalyst under acidic conditions. We could equally do the top arc of this cycle. So the reduction of oxalosuccinate to isocitrate, its dehydration to aconitate, and its rehydration, but on the other carbon, to citrate. And that required the combination of metallic iron as reducing agent and zinc 2 and chromium 3 as co-catalysts, again under very harsh acidic conditions. So it was clear that the conditions that we found are not really relevant to the origin of life. It ended up being like 140 degrees and 1 molar HCl to get all 6 of those reactions to work in the same pot. But it did show for the first time that multiple reactions along the metabolic pathway could happen in sequence under one set of conditions, and that was kind of a new thing at the time. I'll show you later a version 2.0 of this work that is quite a bit milder in terms of the conditions that we use. So at that time, of course, we wanted to access more of that cycle, or at least more of the metabolites in that cycle in simple ways. And so we got interested, but we had struggled to get the ATP-dependent reactions of that cycle to work without enzymes. We tried different phosphorylating agents that might mimic what ATP does, and we just didn't see the reactivity in our experiments. I think that might be for multiple reasons, which I won't go into. But we became interested by this paper from Segre and coworkers that showed that there is a subset of reactions within biochemistry that appear to be ATP independent. And they include the compounds I've circled in black are what I described previously as these 5 universal building blocks for metabolism. They're all present in this network. And the green dots here represent the compounds. The gray lines that interconnect them represent metabolic reactions. And the size of the dot represents how branched that compound is within the network. And if you look at the compounds that are based only on carbon, hydrogen, and oxygen, the 2 biggest branching compounds are pyruvate, which I just showed you we make from CO2 and hydrogen, and galoxylate, which is a reactive C2 aldehyde in biochemistry. So this suggests that maybe if there was a metabolism that functioned without ATP at one point, or a subset of it anyway, that it would have depended heavily on those two compounds. And at the same time, there had been a paper that had just appeared from Springsteen and Krishnamurthy's groups, and they had shown that you could do an aldol reaction between galoxylate and pyruvate without any catalyst at all. And subsequent reactions could also occur under different conditions. But this inspired us to think, what would happen if we looked at simple geological catalysts like iron, for instance, in the presence of those compounds? So we just mixed together galoxylate and pyruvate in the presence of a panel of different ions and minerals and just saw what happened. And I think we were blown away to see that in one experiment that used ferrous iron as a catalyst at 70 degrees Celsius, we started to see all of a sudden 9 of the 11 Krebs cycle intermediates appear as we could detect and quantify them by GCMS. The reactivity was not exclusive to iron. It worked with other divalent metal ions that we tried. Most of them worked, although simple HCl or potassium chloride were not effective catalysts. So this was interesting, and I'll show you what the reactions that are happening. We tried to piece them together by a couple of approaches. So one is just to sort of resubject all of the intermediates that we saw back to the reaction conditions in the absence or presence of pyruvate to see how they progressed. And we also used C13 labeled galoxylates and pyruvates. And using those labels, we could follow it around with the mass spec and see how it got transferred around. That gives us an idea of the chemical reactions that might have happened. And so we determined that the first reaction in the sequence is, as Springsteen and Krishnamurthy had shown, is an aldol reaction to give what we call hydroxyketoglutarate. And then subsequent to that, there's a dehydration to give oxopentene dioate, and that double bond is then reduced to give alpha ketoglutarate. Alpha ketoglutarate now can do another aldol reaction with galoxylate, and that gives us this compound, which is a 1-carbon homolog of isocitrate. In fact, it actually gives us isocitrate once it undergoes a reaction known as an oxidative decarboxylation. And similarly, these other keto acids that are generated by this process, they also undergo an oxidative decarboxylation to give us succinate, fumarate, and malate that are all intermediates of the Krebs cycle as well. So this is how these Krebs cycle intermediates are forming. They're forming by aldol condensations in sequence with a reduction reaction connecting them. And that turns out to be actually the slow step of the process. Later, Krishnamurthy and Springsteen showed that it's galoxylate that is doing the reduction of this double bond, and that seems to be the bottleneck of the whole process. So they also showed, in fact, that you could use phosphate buffer as a catalyst, which is an acid-base catalyst, rather than the Lewis acid catalyst that we'd used in ferrous iron. And so we also showed that you can convert those keto acids into the amino acids if you add in hydroxylamine and metallic iron into the experiment at a specific time. So that's kind of cheating in the sense that you have to take an action at a specific moment. But as I'll show you later, we've now gone over that as well. So at that point, I think we started to hit a limit of what we could do with this sort of chemistry using this approach. And just when that happens in science, usually it's a sign that some of your assumptions are probably wrong. So at that point, we went back to the drawing board and asked ourselves, what were our assumptions going into this? And probably a key assumption was that, and this came from especially the papers of Vectorschauser and others, that, as well as Bill Martin and Mike Russell's papers, that if today that you have an enzyme doing a certain job and it's based on, let's say, an iron sulfur cluster in its active site, right? Like take the hydrogenases as an example, that previously the original catalyst that may have enabled this was a mineral or that may have resembled structurally the active site of that enzyme. We've even broken that down in the examples I just showed you to include things like ferrous iron and metallic iron. And that there had been some success along those lines, but we were limited in what we could eventually achieve. So maybe that assumption is not true. Now, what other way can we look at that question? Well, let's consider how humans discovered catalysis. If you look at the hydrogenation reaction, which is of a carbon-carbon double bond, it's a very common reaction done by chemists all over the world, and especially in industry. The first catalysts discovered for that process were not based on abundant metals. They were based on very rare metals like palladium or like rhodium. And those are excellent catalysts on their own. You don't need to do anything to them to make them a good catalyst. It takes a long time for chemists to realize that you can use slightly more abundant metals like ruthenium or like cobalt to do this chemistry. But then you have to do something to them. In the case of ruthenium, you have to convert it from ruthenium-3 into ruthenium-2, and then you have to stabilize it some way with ligands. Same goes for cobalt. You have to add some ligands to make it work. And iron, which is one of the most abundant metals in the Earth's crust, it is uncovered into the 2000s as a catalyst for this reaction, when Chirk realizes that you can have to put a very complex organic molecule around it, we call it a ligand, to modify the energies and properties of the intermediates in a catalytic cycle such that the cycle can turn over. And we asked ourselves, why would prebiotic chemistry not have to go through the same progression? So, if rare metals are excellent catalysts on their own, even if they're rare, could it be that you actually have to use them to initiate a process, and then as the organics from that process become ligands, they can bind to more common metals, turning them into good catalysts? And that's how we end up with the iron sulfur-based catalyst that we have today. Now you might, so what I'm saying essentially is that perhaps the earliest ancestors to some of these iron hydrogenases, for instance, aren't iron-based at all. Maybe they're based on very rare but very reactive metals like palladium and rhodium. Now you might think, okay, that's crazy, because palladium and rhodium are present on the early Earth and on the Earth's crust in parts per billion levels. So that's an issue. Maybe they're about 1,000 times more concentrated in meteorites, for instance. But there's also natural processes that concentrate them. If you go to Russia, you can find rocks that are 11% palladium by weight. Okay, so even though they're very dilute or low concentration overall on average, there are specific places where they can become concentrated. And also the catalysts and reactions we're talking about are ones that are based off of gases that are ubiquitous. So wherever these metals are concentrated is where the reactions could happen. So what happens if we now revisit this reverse Krebs cycle, taking in the possibility that maybe some of these rare metals might have catalyzed them, or maybe even some not so rare metals? So we found in the case we did a screen, this is Sophia's work, recently came out on Gavana, that she found that, well, some abundant metals like nickel can catalyze this reaction, but you need not so much of it, but some of it. There's actually catalytic turnover. We can see a number of turnovers in those systems. Or if you use rhodium, you can use amounts as low as 32 parts per million, and you can get hundreds of turnovers in this reaction. She's using hydrogen gas now to power these reactions instead of metallic iron, and now they work at neutral pH. They work at acidic or alkaline pH, pretty much any pH we tried. They work at room temperature, and they can even work anywhere above the freezing point of water. We did them at 5 degrees Celsius. They work perfectly fine. We submitted the paper for publication, and the reviewer suggested, oh, it'd be nice to see a bit more geologically plausible catalysts. And Sophia had the sense to obtain some samples of meteorites, and she got 3, in fact, initially. Now we have many more. And she tried the Campo del Cielo meteorite, the Gibeon, and the Cicote Elin. I hope I'm pronouncing those correctly. I don't come from that field. You can correct me at the end. But it turns out that after she ground them up and put them in the reaction, they all work. She showed through control experiments that if the hydrogen isn't present, the reaction doesn't happen. So it's not the meteorites that are the reducing agents, it's the hydrogen gas. And the reaction is very clean and works very well. So, okay, that's rhodium. What about other rare metals? Well, Emily and Harpreet and Jing in our lab investigated those. If you try to mix hydrogen and CO2 in the presence of small amounts of palladium, just 32 parts per million, which is equal to 0.1 mole% relative to the bicarbonate that we put into a system, you hydrogenate at room temperature and pH 8 bicarbonate to formate in very good yields. We got more than half of that bicarbonate becomes reduced to formate, and there's no other product. It's very selective. So hundreds of turnovers again. which is pretty rare in the field of prebiotic chemistry. It's not the only chemistry you can do. Now, if you I should have updated this slide. A few mistakes here, but anyway, if we now revisit that same chemistry that happens between glyoxylate and pyruvate, remember I mentioned that previously the slowest step in that chemistry was the reduction of this carbon-carbon double bond. And that was done previously by glyoxylate at elevated temperatures in the presence of large amounts of catalyst, either 200 mol% of iron in our case, or 500 mol% of phosphate in the case of Krishnamurthy and Springsteen. Now this double bond is reduced at room temperature using 64 parts per million of palladium, so 1 mol% of catalyst. And it works at room temperature without any buffer and at neutral pH. And it's higher yielding as well because we can recover more of the mass. We're not working at high temperatures and destroying the material. So that's interesting. How about what can you do with keto acids? Well, one of the things you can do is turn them into amino acids, just like biochemistry does. Biochemistry converts these into amino acids by reductive amination, in the case of ketoglutarate, or by transamination, which means you already have an amino acid and you transfer its amino group or ammonia to another keto acid. And you especially need glycine and aspartic acid because those are the amino acids that are the building blocks of ribonucleotides in terms of their carbon skeletons. And so can we mimic that chemistry? In fact, we can mimic both of those processes, as I'll show you, without enzymes as well. So it turns out with this palladium and hydrogen gas, or instead of hydrogen gas, we can also use formate, which I already showed you is produced by hydrogenating bicarbonate, that we convert quite efficiently these keto acids into amino acids. And it's not working just for one or two keto acids as in previous work done in the area in prebiotic chemistry. It's working for all of the keto acids on this screen, which is pretty much all of the relevant, biologically relevant keto acids. It's interesting because if you use hydrogen gas alone without the bicarbonate or without CO2, you get actually not the amino acid predominantly, but the hydroxy acid. And we interpret this as either that the CO2 is assisting in the reaction mechanism or that it's actually initially producing formate, which then acts as a transfer hydrogenation agent to produce the amino acid. That's why we can use formate on its own without the hydrogen, and it still works and gives us very similar results. Now, the interesting thing about the last three slides I showed you is that the output of one process is the input of another. And so can we then combine all these three functions to work in a single flask? And the answer is yes. So Harpreet mixed oxaloacetate and glyoxylate in the presence of bicarb and hydrogen and ammonium at room temperature and neutral pH. The same aldol chemistry happens with the reduction aided by the hydrogen gas. It then undergoes reductive amination all in the same pot, and we predominantly get out of that mixture aspartate and glycine out of the various amino acids that could in principle form, and in a decent mass recovery of the total compounds that are formed. So this, I think, is one of the larger coherent reaction networks that happens all in the same pot and that mimics biochemistry. Transamination, as I mentioned, is the other way of making amino acids, and we've showed that nickel-2, copper-2, vanadium-5, and cobalt-2 can all catalyze this reaction relatively efficiently in terms of this example between glyoxylate and glutamic acid to give us glycine and alpha-ketoglutarate. Here you can see the rate of the uncatalyzed background reaction, and you can see it compared to the reaction progress in the presence of 20 mol% of these various metal ions. And in the case of copper-2, we have a 2,400-fold rate acceleration compared to the uncatalyzed background reaction. So that really opens up a new possibility for us. In fact, it allows us to pose the question, or to use transamination as a case study for how biocatalysis might have evolved over time. How did organic chemistry and these reaction networks produce their own catalysts rather than relying on catalysts that were just naturally present in the environment? So I've just shown you that these four metal ions are quite efficient catalysts for transamination on their own. We know that today that these reactions are catalyzed without metals. They use a cofactor known as PLP, pyridoxal phosphate. They also use an enzyme as to donate a proton that actually activates the PLP. So the question is then, if those two things were connected, there should be intermediate states in which we can use maybe metals and the cofactors together. Maybe the cofactor emerged and made the reactivity of some metals better, and that could explain why it was retained, and then eventually enzymes replaced the metal, and that's how we get to the system that we have today. So does such an intermediary state or evolutionary state exist? And so what Quentin did is he took the reaction of the transamination reaction between pyruvate and glutamate. He measured the rate in the presence of PLP with a specific catalyst loading, and he sets, let's call that rate, we'll set that relative to 1. And then he measured the rate of the reaction in the presence of the same cofactor, but now in the presence of an equimolar amount of various metal ions, all the metal ions that are shown down here, and they're arranged from left to right by order of abundance, with iron 3 and aluminum 3 being the most abundant. And what he found was, okay, well, in the case of the metals alone, here are the rates. Here's the rate. Here's the rate equal to 1, right, for the reaction that happens without any metals, with PLP alone. And for those metals that were already working on their own, we find out they're accelerated by PLP, or pyridoxal, and PL in this case, by 26 and 46-fold in the case of copper and vanadium, respectively. But in the case of many other metals, oh, sorry, these are the metals on their own. They're already 26 and 46 times faster than PL. But if you now combine PLP and those metals, it's now 98 and 79-fold respectively faster than PL on its own. But what's interesting is if you extend that screen of various metals to include metals that were not so good before, such as iron 3 and aluminum 3, you can see here are not very good when they're just on their own. But in the presence of PL, they are accelerated by 85 and 90-fold respectively. So now they're essentially as fast, roughly, as the reaction of copper or vanadium in the presence of PL, and they're faster than the metal catalyzed reactions alone, and far faster than they were without the PL. So we're going from a reaction that could have initiated with relatively rare metals. They're not that rare, but not as rare as palladium, but they're still rare. And now they can start working with the most abundant metals like iron and aluminum if the cofactor is present. And so now that can allow us to propose, in fact, indeed, that we may have started this reaction with more rare metal ions, progressed through a state, once we had a reaction network that could produce our cofactor, the cofactor binds very abundant metals and turns them into catalysts that are even better than those rare catalysts. And now, if that's done for many reactions, the reaction network could spread from an initial environment that only involves rare metals to one that has more abundant metals. And eventually, you produce enzymes that are now replace that metal, and now the reaction network uses only catalysts that it makes itself to catalyze its reactions. Now, I didn't talk about sugars. I'm not going to spend much time on that. I just want to mention that Marcus Rauscher has published papers on a non-enzymatic glycolysis. That's the process by which sugars are broken down. Our lab has been studying how you can build up sugars by roughly the reverse process known as gluconeogenesis. We've gotten most of the reactions to work without enzymes as well, but they seem to work under different pH conditions. And so I think that's notable. But one of the things that Rauscher found when he started with more complex sugars is that they can produce ribose 5-phosphate, which is interesting because it's the building block for ribonucleotides. So let's can we make ribonucleotides? I'll just show you how nature does it. It starts with carbamoyl phosphate, which is just generated from bicarbonate and ATP and ammonia, which comes from amino acids. And then you react that with aspartate to get a carbamoyl aspartate. It cyclizes, releasing water to give dihydroorotate. It's oxidized, this carbon-carbon bond, to give orotate. And then orotate combines with an activated form of ribose 5-phosphate known as PRPP, or phosphoribosyl pyrophosphate, and that gives us orotidine monophosphate. This is not one of the canonical nucleobases. Before you get the canonical nucleobase, which is uridine monophosphate, you have to decarboxylate this group, losing CO2, and then you get uridine monophosphate. It goes on later to form to form cytidine, one of the other canonical ribonucleobases that is a pyrimidine. So can this process occur without enzymes as well? Jing looked at this. It turns out that it can. And so she used the The first reaction happens just without any catalyst at all at pH 8. It can happen even at room temperature, but she showed it happens at 60 degrees as well. The cyclization to give the 6-membered product, the biological pathway does occur, but it also is co-occurring with the one that gives you the 5-membered product, hydantoin 5-acetate. But when you oxidize this compound to orotate, we found that hydrogen peroxide could do it, or manganese 4, manganese dioxide, works better in the presence of copper, although copper is not required. Those same conditions also oxidize this compound if you use manganese dioxide, and then under alkaline conditions, it rearranges to give you the biological product as well. So that's an interesting thing. So maybe the take-home message here is that yes, this pathway may possibly operate with the enzymes. It probably involves pH fluctuations, as we observed. Maybe originally it was not a linear path, but it actually had branching points, which ultimately ended at the same place. And over time, enzymes got selected that prioritize one pathway over the other, possibly because it was more direct. I don't know. In any case, we were able to do this chemistry in one pot, but we had to change the pH. So maybe this implicates pH gradients that could be involved for this sort of chemistry in some sort of natural environment. Okay, so this is the last thing I want to say. You know, we talk so much about ribonucleotides and RNA, but you know, why did ribonucleotides emerge in the first place? I mean, nature didn't know it was going to make RNA when it made them, and so if you ask, your answer to this question may depend on what sort of philosophy that you have on the origin of life. And if you follow a genetics first approach, which is that some prebiotic chemistry that is probably distinct from metabolism gave rise to ribonucleotides, and then they polymerized and gave polymers with some sort of function. Maybe they could self-replicate, maybe they were catalysts. Then you don't really care about the function of a ribonucleotide. What you care about is the function of a polymer. But in the metabolism first hypothesis, ribonucleotides could only survive within like any other small molecule, would only survive within a network if they had some benefit on the self-organized network, if they reinforced their own synthesis or they stabilized the synthesis of the whole network in some way. So we would expect function from a single ribonucleotide in some way, but none is yet known as far as I know. So we became interested in a paper from two papers from Catani in the early 1990s, and more recently from one by Lane. In fact, the student, the postdoc who did this work that I'm going to show you actually had done her PhD with Lane and published this paper during her PhD. Essentially, in the previous work from Catani and by Lane had shown that you can take ADP in the presence of an acyl phosphate and iron 3, and it will transfer the phosphate group from the acyl phosphate to ADP to give ATP. And ATP, of course, is commonly referred to as the universal energy currency of all life. But that process was found to be really specific. So if you used any nucleobase other than ADP, if you used GDP or CDP or UDP, the reaction didn't work. So we wondered if the ADP may have had a double role. If it wasn't just only good at receiving phosphate, it was also a catalyst for that phosphate transfer. And so we decided to do the same reaction, ADP in the presence of acetyl phosphate and iron 3, but now add in an external nucleotide or nucleoside diphosphate, one of the ones that did not react originally. And all of a sudden, we see the reaction happens in those as well. So that indicates that ADP, and in working together with iron, is actually catalyzing phosphate transfer, phosphorylation from acetyl phosphate to the various nucleotide diphosphates to give all of the canonical nucleoside triphosphates as well as ITP. So this reaction only works with iron 3 and with aluminum 3. We tried many other metals, none of them work. Only ADP acts as the catalyst, none of the other nucleotide or nucleoside diphosphates work. But what this shows is that individual ribonucleotides can have a larger function within some sort of proto-metabolic network. They can produce NTPs, all of the canonical NTPs, which are very important because those are the building blocks that go on to polymerize to give RNA, that are the biosynthetic precursors to other ribonucleotide triphosphates, and that are also the biosynthetic precursors to deoxyribonucleotides. So these are very important. This is a very important process, and it is catalyzed by a simple ribonucleotide and a metal ion. This may also help to explain why ATP is actually used as an energy currency in biochemistry. And these adenosine diphosphates are good catalysts, and the products they produce can then be used to further transfer phosphate to other things. So it's nice if you have your phosphate transfer catalyst built in to that function. It also links a function done by a simple small biomolecule to something done by a very large one. So we know that today, when the process by which nucleoside diphosphates are phosphorylated involves the use of ATP as phosphorylate donor. You get your NTPs. It is catalyzed within an enzyme using a histidine residue. Histidine's biosynthesis comes from the breakdown of adenosine derivatives. So and I've just shown you that the same job can be accomplished just by a simple ADP plus iron. And so this, and of course, this might be a biosynthetic precursor to histidine. So you could see this process beginning in this way. A byproduct of histidine adenosine catabolism leads to histidine, which then does the reaction better than the original process, and then it takes it over. You could actually see how that progression might have played out in evolutionary history. Okay, so that brings me to the end. We've overviewed many different processes in the core of metabolism. We found that many of them can react and occur without enzymes. There's much more work to be done. There's lots of processes that we haven't yet seen, and we have to link it all together in one environment. But we're getting lots of clues on how that might have happened now from collectively looking at all of these processes. So my conclusions are that the chemistry that gave rise to life must have been self-organized. That's pretty much got to be true. It was probably driven by redox agents and activated phosphate. It was probably catalyzed by metals and acid-base catalysts naturally present in the environment. I think now there's ample evidence that at least a lot of this chemistry can actually happen that way, as I've just shown you from our group and from others. I've introduced the idea that rare metals that are not used by biology today may still have been relevant catalysts at the beginning, and then been replaced by more abundant metals once organic complexity had increased to a certain level. And I've illustrated that by showing that some cofactors may have initially been ligands that accelerated catalysis by metals of abundant metals that originally were done by those rare metals. So, what are the major frontiers moving forward? For sure, prebiotic phosphorylation is a big one. And it's a big question for pretty much everyone working in the origin of life field. And I talked about constraints, and I've mostly looked at catalysts and conditions as constraints. I haven't looked at gradients. We haven't looked at scale as being another important constraint. All of life's chemistry happens on the micron scale, right, within cells. And of course, integrating all of these into even more into common environments so that the whole network can function as a whole is the ultimate goal. And that will hopefully, come in sometime in the future. So I got to thank the people who did this work. Of course, it wasn't me that did any of it. I already showed the names of all the students and postdocs who had physically done the experiments. I also have to thank Camilla Muhauska, who was a CNRS researcher in the group, and also was a postdoc, contributed a lot to the work, especially in the beginnings. This is a photo, not of my current group. It's actually from a 10-year lab party, 10-year anniversary lab party, where we invited a lot of former coworkers to come back and have a big party together. They could tell us about their careers in industry and in academia and in government, and that was a lot of fun. I got to thank our collaborators, Bill Martin and Harun Tusuz in Germany, who've been fantastic collaborators, and I've got to thank many funding agencies, as we all have to do, especially the ERC, most importantly. They funded essentially all of the work I showed you. Marie Curie Actions for funding an ITN and postdocs that we benefited from, notably the proto-met ITN network that's now coming to an end, and the Excellence Cluster in Strasbourg and the Volkswagen Foundation for support. Thanks again for inviting me to this lecture. I'm happy to answer any questions that you might have.
Hi, hello.
Hello.
Hello, Victor, please.
Hello,
Yes.
hello, Joseph. Thank you very much for your talk. I enjoyed a lot. I like very much your presentation and the things that you have done is I think it's amazing. I have just a question. What I haven't seen, or I don't know if you have done some comparison between the different kinetics between the catalysts, metal catalysts and corresponding enzymes. What do enzymes add to these reactions, or how do you, or what is the improvements that the enzymes add to the well, let's say primitive biochemistry? And you know how, because here's the point, is there any, what are the extra value that enzyme add to this reaction? Because apparently there is a lot of reaction that metals can catalyze, and somehow one say, okay, we can live with just with metals, no? But this was not the case. So can you tell us something about this?
Yeah, I mean, we've talked about that in the lab. It's a difficult comparison to make, because usually when you benchmark catalysts against each other, you benchmark them on a molar, you compare, the same number of moles of one to another. But one must wonder whether this is a good comparison. So certainly if you do it that way, the enzymes will probably outstrip metal ions by many orders of magnitude. Mm-hmm. On the other hand, the biosynthesis of an enzyme is incredibly energy intensive, right? You might have, I don't know, 10,000 amino acids or whatever in most complex ones, right? And so that requires a lot of energy to make. So even if you have and also the catalyst itself is very heavy. So if you benchmark them on a molar level, I don't think it's really a fair comparison. So maybe you have to benchmark them according to mass or in a different way. Because if you have a high concentration of metal ion, you might have, 1,000 times more metal than you have enzyme by moles, but by mass it might be lighter. So it's a hard comparison to make. I don't know if it's a fair comparison really to compare 1 mole of 1 mole percent of an enzyme versus 1 mole percent of a metal ion, but it's an excellent question, and one that requires one that must reflect on. Of course, there must be a great benefit to making your own catalysts, first of all. But the one thing I don't, maybe other people have thoughts on this, but why do enzymes really have to be so big? Because I come from the field of organic chemistry. People have now made organic catalysts that are relatively small. You can have a single amino acid proline be a pretty effective catalyst for a lot of aldol reactions, for instance. Groups like Scott Miller in Yale and Helma Wennemers at ATH, they've shown that simple peptides can do very, very, very efficient and even enantioselective chemistry with only, 3 to 10 amino acid residues, right? So why are most enzymes so much more, so much more large? There must be a massive benefit to that I don't really understand, because
sorry to interrupt you. Could it be related with compartmentalizations, and how you mentioned in the first slide or the second slide that for sure compartmentalization is a critical aspect for life, and could it be a first kind of compartmentalizations, and how enzymes are big and they somehow bring together the catalytic side to other places, and enzymes at the same time can accumulate, make some partitioning? Could be this what are the reason for?
Yeah, absolutely. I mean, in general, a trend you see in evolution is often you see there's a specialized enzyme for certain reactions, but you can that makes a tunnel between one process and another, so that the same process occurs in the same enzyme but just in a different part of it. But that originally it may have been 2 separate enzymes. But another trend that you see is that some enzymes may have they were maybe generalist enzymes at first. They catalyzed a large variety of processes and became specialized later, so that maybe they got each one original ancestral enzyme was catalyzing multiple processes, but then became specialized for each process over time, and essentially became two distinct entities. If you look at it that way, if you take that to the extreme, you could imagine one universal catalyst that essentially provided an environment for many, many reactions, that's something that's very interesting to think about. People are looking at coacervates as catalysts, or at least compartments now. Dora Tang, for instance, but other people as well. So yeah, I mean, that's a very important question, and I don't know if I'm the best place to answer it, but I definitely think about it and would be happy to collaborate with the right person to try to answer it.
Mm-hmm. Okay, thank you, John.
One thing what actually enzymes have an advantage of is that relates to their size, maybe is that they are very, very specific. So an enzyme can only catalyze normally one very specific reaction. On the other hand, transition metals, they can catalyze quite a lot of different reactions. Now, so now in early evolution, this might not really be a problem if the same enzyme forms maybe some peptide or some integrate some carbon dioxide into a metabolism, and also can at the same time oxidize sulfur dioxide to sulfur trioxide. I mean, this might not really be a problem in the early evolution, but in a modern body, in a complex organism, this might actually be a disadvantage. So you want probably in a complex organism, you want probably something very, very specific. Whereas in early evolution, the specificity
that's exactly. That's a very good point, Wolf. Yeah, sequestering the enzymes may have, you get rid of that background reaction, that non-specific background reaction that you would get in the presence of metal ions, and you allow things to be more selective over time. And certainly, the more complex the system becomes, the more important that becomes. So probably, the minimal metabolism, like the one I depicted on that slide, was probably, well, that's relatively simple compared to the larger metabolism that we have today, and so may not have been. Some of the specificity may have been inherent in the reactions themselves. And one point I can make on that point is, one of the legitimate criticisms of this idea of self-organized metabolism came from Leslie Orgel, and the last paper that he published, it was actually published after his death. And he pointed out that in the specific case of the reverse Krebs cycle, one of the problems would be that some of the reactions that occur on one type of functional group would be non-specific with the catalyst, and that you would get more matter bleeding off of the cycle than would remain on it, or at least equal amounts, or not drastically different amounts. And that would be a big problem for maintaining a cycle that was autocatalytic, because if matter flows off of it faster than it comes in, then you're kind of out of luck. But what we showed in a recent mechanistic study that appeared earlier this year in Angewandte Chemie, if you just compare the rates of reduction of oxaloacetate and pyruvate, which are the two examples that he took, just with sodium cyanoborohydride. This is a common reagent that organic chemists use all the time, and you do it at somewhat acidic pH, like pH 4. At that condition, you get 85-to-1 selectivity for the reduction of oxaloacetate over pyruvate, which is exactly what you need to stay in the cycle. Now, why do you get that selectivity? It turns out that at slightly acidic pHs, the carboxylate of oxaloacetate is protonated, and that provides an intramolecular catalyst, which is a 6-membered ring hydrogen bond, onto the carbonyl that needs to be reduced. So one molecule has a catalyst built into its own structure, and the other one does not, and that's why it's reduced 85 times faster. And there was no catalyst used at all in this process. It was just a polar reducing agent, which could be anything, right? So there may be intrinsic explanations within organic chemistry that govern selectivity in networks at the very primitive stage that we may have not sufficiently appreciated until they're studied in detail. And so thereby, I'd say dismissing it out of hand without investigating it is to be avoided, I would say.
Thank you.
Okay. Are there any more questions or discussion contributions? I don't see anybody, so I would like to thank Joseph again for accepting our invitation. Thanks again. And thanks all for coming, and see you then in 2 weeks.